to soil microbiology lab of IITA. Let me take you around and show you the activities that are being carried out in the lab. First, our priority is the mandate on nitrogen fixation, using biological means to fix nitrogen into the soil. This is a flow hood, a sterile environment where all the strains, all the Rhizobia and so on are being cultured. After culturing them, we move down and keep them in the incubator. We keep them inside this incubator at 28 degrees. After 24 hours, we check the growth. Then if we are satisfied with the growth and we don't want them to overgrow, we keep them in the fridge. These are the fridge where we keep them so that it will retard their growth. Most of these strains, when we have confirmed that, yes, we have these strains, we keep them for a long time for customers or other researchers to come for them. Don't back up your research with molecular characterization or molecular studies. It will not be acceptable. For example, this is where we do our molecular studies of all the bacteria generated from that first point I showed you. So, in soil microbiology, we still work on phosphorus. At that end, we still work on phosphorus, we isolate P-solubilizing bacteria and so on. Here we study mycorrhiza. Mycorrhiza are everywhere in the soil, around the world. But the beneficial one that we are researching on are the vesicular avascular mycorrhiza that develop a kind of hyphae. The spores develop a kind of hyphae attached to plant and help those plants to spread their root to tap phosphorus that is around them. So here we have samples from the field and from some experiments whereby we start to study on the microscope whether the roots are infected or not. So to the next side of the lab. Second side of the lab, where we do all the physical and chemical analysis of all the research from the other side. Most of the strain I told you about up there are to be tested on the field. And testing them requires a situation whereby you come to the lab and do the chemical analysis. Now, for the nitrogen fixation which I discussed at the other time, we have, for example, soya bean. We have different species of varieties of soya bean. So some researchers will demand that, okay, I want to know the one that is fixing nitrogen more than the other. Now, there is a method we use here, which is, we call it urate method. And it has been very successful. Breeders bring their samples. We use the urate method. The urate method will help them to select the best variety of their crop that they can introduce to farmers that will fix more nitrogen into the soil. This is a foam hood. This foam hood, all chemical digestion is being done. And during digestion, a lot of fumes, bad odors used to come out that can affect the health of anybody around. So this foam hood will suck all the odors and the fumes into the air. That is the advantage of this. This is a shaker, vortex mixer. This is a pH meter. And this is a orbiting stirrer. These are soils from IITA from UI brought in by a student for sterilization because they want to use it to conduct an experiment that will involve all these strains that we have been talking about. So they need to kill all the bacteria in that soil so that as to know whether the strains that we are first introduced to you people, whether they are functioning or not. These are urine extracts that are pending for analysis. It has to be kept in the fridge. We still involved in research on microbial biomass because some researchers within the institute and outside the institute used to come to soil microbiology so that we can assess the amount of nitrogen absorbed by the microbes, or the phosphorus absorbed, or the carbon absorbed by the mycorrhiza and released back to the soil. It entails a kind of chemical reactions and so on. So this is what we have been working on and doing in soil microbiology lab.